is also booming, and these are becoming a true option in multiple myeloma. And I'm sure you've seen this kind of slides hundreds of times, highlighting that although myeloma is still an incurable disease in general, we all have a couple of patients who are long survivors, but in general, this is an incurable disease. However, when you consider the standard risk, multiple myeloma, normal beta-2 microglobulin, normal cytogenetics, now these patients are enjoying more than 10 or 15 years survival. This is mainly due because at the time of relapse, we are able to give them a novel agent, achieve some PFS, and putting all of these PFS together, we end up in more than 10 years. However, despite this enthusiasm and excitement, looking recently into this so-called penta-refractory patient, if you have failed proteasome inhibitors, IMiDs, but also the monoclonal antibody daratumumab, definitely this is an unmet medical need. And actually, this is the niche where all of these novel agents are being developed. And we heard also earlier about, for example, selinexor and many other agents. This is also where maybe immune therapy, at least in the beginning, can be investigated. This is a global schema of the different so-called strategies for immune therapies for myeloma, but actually you can apply this to lymphoma or to any other cancer. I will not go through all of them, and my focus will be definitely on the cell therapies. So, like in many other malignant diseases, anti-tumor immunity fail, unfortunately, and we heard about this earlier this morning. So, tolerance, energy, the microenvironment, actually, there are many factors that are contributing to the failure of anti-tumor immunity, and actually we really need to tackle this if we want to achieve success with immunotherapy over the long term. When it comes to CAR T cells in myeloma, the short objective is to redirect the TCR towards the tumor antigen. We would like to overcome HLA restriction and induce an adequate number of high affinity cells. I will not go into all the different CAR T cell constructions. I do not have the expertise to talk about all this, but we'll hear during the meeting about many fancy constructions, and we had during the innovative zone earlier this morning some nice talks about how quickly and how fast these different new generation CAR T cells are being developed. The CAR T cell therapy is a multi-step procedure. You need to identify your specific tumor antigen. You need to produce this high affinity antibody. Of course, you need to genotype the antibody, transduce the autologous or maybe the allogenic lymphocyte. Before going into patient, you need to have a proof of concept about the efficacy in vitro and in vivo. And most importantly, and I think this is a key issue, you would like to show the absence of cross-reactions against a standard panel of normal tissues. And this is why you're not surprised why, for example, CD19 is the most popular antigen, because actually we can live without CD19, whereas the story can be much more complicated if you target an antigen which is shared by the tumor, but also by some important normal tissues. So in multiple myeloma, we do have several potential antigens. CD138, previously called Syndican-1, is a specific marker of plasma cells. The kappa light chain can be also used. I'll show you actually the proof of concept. A few cases looked into CD19, although we know that this antigen is infrequently expressed on myeloma cells. This is highly debated. SLAM F7 is also a candidate, and by the way, you heard about elotuzumab, which is the antibody targeting SLAM F7. However, the most popular antigen that is being currently targeted, and I believe all the experience available, is with BCMA, the B-cell maturation antigen. So let me show you first this initial clinical case report that was published in the New England Journal of Medicine, sort of a proof of concept, although there was some controversy about the expression of CD19 in multiple myeloma. So this is a patient who failed high-dose melphalan, lenalidomide, bortezomib, and actually the patient received melphalan 140, which is actually nice lymphodepleting chemotherapy. Then the patient received the anti-CD19 CAR T cells, and you can see the response rate. Obviously, he received also maintenance with lenalidomide. Of course, it's very difficult to conclude who did what, I would say, whether it's the melphalan, the lenalidomide. Nevertheless, I think it's a nice proof of concept in a patient who failed 9 prior lines of therapy. I think the story is much more exciting with BCMA. This is a member of the TNFR superfamily. It's expressed by plasma cells, some mature normal B cells, but mainly plasma cells, universally expressed in myeloma, but also in some lymphomas. And actually, the good news is that if you take knockout mice for BCMA, they are healthy with normal B cells, but they have reduced plasma cells. So that brings you back to the issue of safety. Looking to our mRNA expression in several cancers for BCMA, you can appreciate here that BCMA is highly expressed in myeloma. Actually, myeloma is the is the tumor where it's the most highly expressed. So it's clearly this kind of figures can allow you to define the patient population of interest for your antigen. So with this background, actually, we do have now many CAR T cells under development in multiple myeloma. These are some of the most advanced ones, but I must confess that this slide is already outdated because during the last ASH meeting, we've seen many other CAR T cells being developed. So I'll show you, it's a choice. Of course, I do apologize if I'm missing your favorite CAR T cell study or CAR T cell construct. So I will try to show you some results, the most convincing results, I would say, mature data. This is the NCI trial published last year in JCO, 16 patients receiving BCMA CAR T cells. These patients received the CAR T cells after conditioning with cyclophosphamide and fludarabine. It looks like for any CAR T cell, cyclophosphamide and fludarabine is really the standard of care. And you can appreciate that almost all of these patients, except one, I think, responded. And actually, the level of toxicity was relatively acceptable. There was maybe a correlation between the toxicity, especially CRS, and the tumor load in the bone marrow. This is a suggestion from different trials and different malignancies. The other interesting experience is this so-called Legend 2 trial using this LCAR B38M developed in China by Nanjing and acquired by Janssen. So this is directed against BCMA. It's an ongoing phase 1/2 study which planned to include 100 patients. We had preliminary findings presented in 2017, about 19 patients, very exciting. And during the last ASH meeting, the authors presented a 12 months follow-up on more patients, 57 patients. And actually, I have seen this paper already in press. So the schema, leukapheresis, they would receive cyclophosphamide. I think there's no fludarabine here. And then the infusion of the CAR T cells. Primary endpoint is safety, and obviously secondary endpoint, anti-myeloma efficacy. I will not go through all of these details of the 57 patients, but definitely these are heavily pre-treated patients. Also, you would see that they did not receive the novel, I would say, agent and the monoclonal antibodies. Still, they have failed a median of 3 lines of therapy. And what you can see, actually, in contrast to previous experience, there is no grade 4 CRS. 4 patients out of 57 developed a grade 3 CRS. Median time to onset is 9 days, and this is now becoming more and more well established, something around 1 to 10 days. Median duration was 9 days for the CRS, and it was successfully managed with the anti-IL-6 receptor tocilizumab. Looking into the response rate, this is actually quite amazing, especially when it comes to MRD negativity. I mean, in myeloma, we never see such high level of MRD negativity, even when you treat patients at earlier stages. The median duration of response was 16 months, and the median duration of MRD negativity at 22 months. And the median time to the initial response was very quick, around a month. So these data are really very appealing, and apparently there is little correlation with the level of expression of BCMA. You can see here the PFS curves for all patients, but also for the patients who could achieve MRD negativity. Definitely a high level of MRD negativity. Still, patients with MRD negativity continue to relapse. Then one needs to question what is the relevance of this MRD negativity in these patients. Looking into the overall survival, these are really very interesting data. And when you look carefully to the causes of death, actually, none of them is directly related to the CAR T cells, actually. So very positive and interesting data. The trial is still ongoing, and I believe that the company is willing to file with these data. The other mature and quite convincing data are the ones with the famous BB2121 from Celgene. And again, this is the schema of this trial, a dose escalation phase followed by a dose expansion, again directed against BCMA. Looking into the treatment history, definitely this is a patient population which is much more heavily pre-treated. You can appreciate 7 or 8 lines of prior therapy, including proteasome inhibitors, IMiDs, but also daratumumab, and of course, autologous stem cell transplantation. So the tumor response was dose-related. So the more you give CAR T cells, actually, the more response you would achieve. However, like in the LEGEND trial, that was independent of tumor BCMA expression. But one must admit that BCMA is really highly expressed in multiple myeloma. Looking into the progression-free survival, also these are different populations, different products, different contexts. Actually, it looks like that the PFS is around 12 months in this population. So you can achieve 12 months PFS with CAR T cells directed against BCMA in multiple myeloma. I kindly remind you that pomalidomide was approved in relapsed refractory myeloma with less than 3 months PFS. So this is something of great importance, especially if you consider the MRD negative patient, then you are around 18 months. We're not yet at the level of cure, but definitely it's an important step. Another question which is of concern in the community, not only in myeloma, but also in leukemia and lymphoma, is about the expansion and the persistence. And it looks like that with this BB2121 construct, you have a CAR T cell expansion with long-term persistence. Of course, what is the definition of long-term? Is it 6 months, 12 months, or for life? So many open questions. When you look to the adverse events, again, it looks like that in myeloma you have less CRS compared to acute lymphoblastic leukemia. Maybe this has to do with the tumor load. Again, no grade 4 CRS and no fatal events, especially CRS or neurotoxicity. And all of these CRS were mostly low grade and manageable. Of course, there's also a learning curve. The more you see, the more you know how to manage, how to prevent, how to anticipate. So the conclusions from this BB2121 are also very positive, I would say, with the median PFS of 1 year in the dose escalation cohort in many patients achieving MRD negativity. And the follow-up trial for this is the so-called KARMA trial, which will aim to include 94 patients. The CAR T cells are not only being developed in the refractory setting. Actually, the field is moving really very, very fast. And Professor Dapeu from mainland China in Suzhou kindly provided me with his slides from the presentation they had during ASH. I didn't alter these slides. This is why I kept them as they were sent to me. So the interesting part of this story is that they have used the anti-BCMA CAR T cells actually in the frontline setting. And this is based on their initial experience actually in relapsed refractory AML. They tested them for safety considerations in 10 patients and actually moved rapidly into the frontline setting, but in high-risk patients. And we know very well that these high-risk multiple myeloma, like deletion 17p patients, are really an unmet medical need. So definitely it is an opportunity to investigate novel approaches. And you can appreciate what they did actually. They infused both CD19 CAR T cells and BCMA, you can see after autologous stem cell transplantation. So a sort of a consolidation therapy. Remember, please, our standard of care is to give auto transplant, consolidation, and maintenance until progression. And it would be really a major step, actually, if we are able to get rid of the minimal residual disease immediately after auto transplant by a single infusion of CAR T cells. So I think this is really a very meaningful and very thoughtful approach. And you can see that in these patients, they could achieve, of course, these are 10 patients. The problem we are facing with CAR T cells, but we are all here to blame, that we're communicating only on very small series. But on the other hand, this is how you learn. You need to go into all the nice stories started with a clinical case. I remember the story about Gleevec. This was, I think, 14 patients, and we know the end of the story. So 10 patients here, a lot of stringent CR. Half of the patient achieved stringent CR. While imagine you would need like 2, 3, 4 years of lenalidomide to achieve this kind of response. You can get it here with one infusion of CAR T cells, and you can see the expansion of these CAR T cells and their persistence several weeks after infusion. Of course, with all the limitations of this kind of comparison, and I'm not a statistician, nevertheless, in their experience compared to historical data, these results are much better. And actually, when you look to the results in the front line compared to the refractory setting, it looks like in the front line you have a better expansion. And this may have to do with the use of autologous, with the use of high-dose melphalan, because after high-dose melphalan, actually the lymphodepletion and the cytopenia environment is very favorable to the expansion of CAR T cells. And we discussed before lunch, for example, IL-15. And actually, you have a lot of IL-15, I believe, after high-dose melphalan. So this is really a fascinating story, not only in the relapsed refractory setting. CAR T cells are now moving into the front line quickly, thanks to our Chinese colleagues who are really being innovative in this field. Of course, we need to improve the tolerance. Also, no patient died of CRS in these different trials, but still, the management of these patients is quite critical. It's not a pill that you can simply give and then send them back home. So these are several weeks or days of hospitalization. And even if the numbers are small, there are still strong side effects, and we need to learn how to manage them quickly. So they are really highly attractive, these CAR T cells in myeloma. I believe after the approval of CAR T cells in ALL, in lymphoma, myeloma will be probably the next candidate. I think we can still improve the efficacy with the humanized construct. The dual B-specific CAR T cells are also now emerging. Probably we need long-lasting CAR T cells. This is why we need to work maybe on the starting material. For the moment, we're using the soup, like, after apheresis. Maybe now selection of the different T cell subset is needed. And I believe more and more that if we really want to optimize the use of these cells, we need to move earlier in the course of disease and not wait until 7 or 8 lines of refractory disease. Thank you very much for your attention.
Thank you, Mohamad.